Hello everyone, I'm glad to share our work on iMeta and the title were Tomato Microbiome on the Long-Term Organic and Conventional Farming. The application of chemical fertilizers have brought serious environmental problems. Organic farming is a green production compared to conventional farming systems. The most famous long-term organic trials were Lawson Experiment Station, reflected the history of agriculture development over the past century, and the results clearly showed that the important contribution of fertilizers, breeding, crop rotation, and plant protection to crop yields. We started long-term trial from 2002 and conducted a comprehensive study of different farming systems on soil micromorphy, nutrition, and microbiome communities. And this study focused on the diversity and dynamics of tomato bacterial communities based on long-term trial. Our main results were as follows. Total nitrogen concentration and soil organic content was higher in the organic greenhouse than that of conventional greenhouse. Bacillus, Bacteria, Actinobacteria was the most prevalent taxonomy both in organic and conventional greenhouse. Alpha diversity analysis revealed that only Simpson index was significantly different between roots and stems, and there were no difference between cropping patterns. Tomato organ compartment and growing phases had significant effects on tomato endophytic bacterial community, and there was a significant difference in bacterial community between roots and stems. However, the difference in endophytic bacterial community of tomato between organic and conventional models were not significant. Lab analysis showed that Bacillus, Symphomargus, and Rhizobacterium were enriched in the organic greenhouse, where Enterobacteria were enriched in the conventional greenhouse. The functional prediction analysis showed that the endophytic bacterial metabolic function groups of tomato mainly included four major groups, such as lipid metabolism and xenotransduction, and there was almost no difference between organic and conventional tomato endophytic bacterial functions. The network analysis in organic and conventional mode revealed that the modularity of tomato endophytic bacteria was higher in the organic greenhouse than that in the conventional greenhouse, and the modular composition was simpler in the conventional greenhouse. And the results of threshold parameters based on network topologic rules revealed that the endophytic bacteria key species were mostly distributed in the phylum Protecbacteria and Firmicutes. Total soil nitrogen content was found to be the major driver of tomato endophytic bacterial community based on RDA analysis, and a positive correlation was observed between majority of tomato endophytic bacteria and soil physiochemical properties. A total of 163 bacteria were isolated by culture-dependent method, but after sequencing alignment to remove reduction, we obtained 60 bacteria from different genera. Firmicutes and Protecbacteria were dominant endophytic bacterial phylum. Specifically, Bacillus were prevalent in the organic farming greenhouse than that in the conventional farming greenhouse. In conclusion, long-term organic manipulation exerted limited influence on the variations of bacterial communities under long-term trial, where compartment was a major driver force for the shifts of endophytic bacterial communities. Organic greenhouse tomato bacteria had more complex co-occurrence networks than that in conventional greenhouse, and root bacteria had more complex co-occurrence networks than that in stem bacteria, respectively. Cultivable method results found that Protecbacteria and Firmicutes are dominant phylum in the endophytes, and Bacillus were more prevalent in the organic farming greenhouse. Thank you for your attention.